Hello there. I'm so glad you found your way here tonight. Wherever you are in the world right now, allow yourself to settle into this shared moment of stillness. Let the noise of the day fade away. Let your thoughts grow quiet, like the slow unfurling of a leaf toward morning light. Right now, as you listen to these words, something extraordinary is happening all around you. In forests, fields, and gardens, a hidden world of awareness is quietly at work. Plants are sensing their surroundings, communicating with neighbors, and making decisions without a brain or a single nerve cell. Tonight we journey into the green world, into the silent realm of plant intelligence. A world of chemical signals, underground networks, and behaviors that challenge everything we thought we knew about what it means to think. We will explore how plants remember past experiences, how they warn their neighbors of danger, how roots navigate obstacles with purpose, and how the boundary between plant and animal intelligence may be far blurrier than we once believed. All of it waiting to be shared in the stillness. Tonight, let's drift off together while exploring over 100 soothing facts about plant intelligence. There's no rush here, no test at the end. Just soft words and quiet wonders to carry you into rest. Welcome to Drowsy Discovery. I am glad you found your way here tonight. Perhaps you have had a long day. Perhaps you simply enjoy falling asleep to thoughts about the remarkable hidden lives of plants. Either way, this space is for you. There is something deeply calming about contemplating the quiet wisdom of the botanical world. Every green leaf is a solar panel, millions of years in the making. Every root tip is an explorer, navigating the dark mysteries of the soil. Every flower is a message written in color and scent. You are connected to this story more than you might realize. The oxygen filling your lungs right now was released by plant life somewhere on this planet. The food that sustains you traces back to photosynthesis. The very atmosphere you breathe exists because of the green world's patient work over billions of years. If you enjoy these gentle journeys, I invite you to like, subscribe, or share a thought below. It helps others find their way here too, one sleepy soul at a time. But for now, all you need to do is relax. So settle in. Let your breathing slow. And let the weight of the day drift away, like pollen carried on a summer breeze. Plants are patient beyond measure. They grow without rushing. They adapt without anxious thought. They communicate in languages we are only beginning to understand. And yet, within all that stillness, there are remarkable acts of perception, memory, and response. Tonight, we will wander through this magnificent green world. We will meet the processes that govern plant behavior and discover abilities that may surprise even the most curious minds. But there is no hurry. Plants have been quietly thriving on Earth for over 400 million years. They can wait a little longer while you rest. Let us begin with the fundamental question of whether plants can truly be said to think. Plants can sense at least 20 different environmental signals at once. Far from being passive organisms simply swaying in the wind, plants are continuously monitoring their environment with remarkable sensitivity. Scientists have identified over 20 distinct environmental parameters that plants can detect simultaneously, including light intensity and quality, temperature, humidity, gravity, touch, soil nutrients, water availability, and the presence of pathogens or herbivores. Each of these signals triggers specific molecular responses that allow the plant to adjust its growth, metabolism, and defensive strategies accordingly. The ability to integrate all this information at once is a form of biological computation that rivals the sensory processing of many animals, Plants achieve this without a centralized nervous system, instead using chemical messengers and electrical signals that flow through their tissues. This multi-channel sensing allows plants to survive in constantly changing environments, from the shifting patterns of sunlight through a forest canopy to the fluctuating moisture levels in soil. The sheer breadth of their environmental awareness challenges our assumptions about what it means to perceive the world. Some plants can distinguish between the touch of rain and an insect. When raindrops fall upon a leaf, the plant experiences mechanical stimulation, but remarkably, many plants can distinguish this harmless contact from the potentially destructive touch of an insect herbivore. This discrimination appears to rely on differences in the pattern, duration, and intensity of the stimulus. Rain tends to produce brief, distributed impacts, while an insect creates more sustained, localized pressure, often accompanied by specific vibrations associated with chewing or probing. Some plants have been shown to respond defensively only when they detect the characteristic signatures of insect feeding, conserving energy by not mounting unnecessary responses to rain. This selective responsiveness is a sophisticated form of sensory filtering that allows plants to prioritize real threats. The mechanisms underlying this ability involve complex signal transduction pathways that interpret mechanical input in context. Such discrimination reveals that plants are not merely reacting to their environment, but actively interpreting it with considerable finesse. The sensitive plant folds its leaves within seconds of being touched. The sensitive plant, known scientifically as Mimosa pudica, is one of nature's most dramatic demonstrations of plant responsiveness. When touched, its delicate compound leaves fold inward, and the stem droops within just a few seconds, a movement rapid enough to startle any observer. This response is driven by changes in water pressure within specialized cells called pulvini, located at the base of each leaflet and leaf stalk. When stimulated, these cells rapidly lose water, causing the leaf structures to collapse. The purpose of this behavior is thought to be defensive, potentially startling herbivores, making the plant appear wilted and less appetizing, or exposing hidden thorns on the stem. The speed of the response rivals that of many animal reflexes and requires the transmission of electrical and chemical signals throughout the plant. The sensitive plant has become a favorite subject for researchers studying plant signaling and has provided key insights into how plants can achieve rapid, coordinated movements. Plants can learn to ignore repeated harmless stimuli. One of the most intriguing discoveries in plant science is that plants can exhibit habituation, the ability to stop responding to a stimulus that proves to be harmless over repeated exposures. In a groundbreaking study using the sensitive plant Mimosa pudica, researchers repeatedly dropped the plants from a short height that caused no damage. Initially, the plants closed their leaves in response to each fall, but after several repetitions they stopped reacting, having apparently learned that the stimulus posed no threat. This is not simply fatigue, because the plants remained fully capable of responding to a new stimulus such as touch. Habituation is a basic form of learning that was long thought to require a nervous system, yet plants achieve it through purely chemical and molecular mechanisms. The discovery challenges traditional boundaries between learning organisms and non-learning organisms. It suggests that the capacity to adapt behavior based on experience may be far more widespread in the living world than previously recognized. A study showed that sensitive plants remembered a lesson for weeks. Not only can plants learn to ignore harmless stimuli, but they can also retain this learned behavior for remarkably long periods. In experiments with Mimosa pudica, researchers found that plants which had habituated to repeated dropping remained unresponsive to the same stimulus even after 4 weeks, without any further exposure. This long-term retention of a learned response resembles memory in animals, and suggests that plants possess mechanisms for storing information about past experiences. The molecular basis of this memory is not fully understood, but likely involves changes in gene expression, protein synthesis, or alterations to cellular signaling networks. This finding adds another layer of complexity to our understanding of plant behavior and suggests that plants may accumulate experiential knowledge over their lifetimes. Such memories could help plants optimize their responses to recurring environmental challenges, conserving resources by not reacting unnecessarily. The patient retention of lessons learned hints at a quiet form of intelligence unfolding in the green world all around us. Plants can detect the direction of light without any eyes. Despite lacking anything resembling eyes or a visual system, plants are exquisitely sensitive to light and can accurately determine its direction, intensity, and even its color. This ability is mediated by specialized photoreceptor proteins found throughout plant tissues, which respond to different wavelengths of light and trigger signaling cascades that guide growth. Stems bend toward light sources through differential cell elongation on the shaded side, a process called phototropism. Leaves adjust their angles to maximize light capture for photosynthesis, while avoiding overexposure that could damage delicate tissues. Some plants can even detect the ratio of red to far red light, which tells them whether they are shaded by other plants and need to grow taller to reach the sun. This light sensing is distributed throughout the plant body rather than concentrated in any single organ, creating a holistic awareness of the light environment. The precision with which plants track light rivals the orientation abilities of many light-sensing animals. Roots can sense gravity and grow downward even in complete darkness. Plant roots display a remarkable ability to sense the direction of gravity and orient their growth accordingly, a phenomenon known as gravitropism. This sensing occurs even in total darkness, independent of any light cues. Within the root tips, specialized cells contain dense starch-filled bodies called statoliths that settle at the bottom of the cell in response to gravity. The position of these statoliths triggers signaling pathways that redistribute growth hormones, causing differential cell elongation and steering the root downward. This gravitational awareness is essential for anchoring the plant and reaching water and nutrients deep in the soil. Experiments in space have shown that plants grown in microgravity lack this directional cue and may orient their roots randomly or display altered growth patterns. The ability of roots to continuously sense and respond to gravity even as they navigate around obstacles demonstrates a persistent, adaptive orientation system. It is a quiet but vital form of spatial awareness that helps plants thrive in their terrestrial environment. Some plants can detect the presence of water before touching it. Certain plant roots exhibit the ability to sense moisture gradients in the soil and grow preferentially toward water sources before actually making physical contact with them. This phenomenon, called hydrotropism, allows roots to locate water more efficiently, especially in environments where moisture is unevenly distributed. The mechanism involves detection of humidity gradients by root cap cells, which then direct growth toward higher moisture concentrations. Remarkably, in some cases, this response can override the stronger gravitropic signal, causing roots to grow sideways or even upward, if that is where water is located. This anticipatory behavior helps plants survive in drought-prone environments by optimizing the allocation of limited root growth resources. Scientists are still unraveling the molecular pathways involved, but the ability to sense water at a distance is clearly a valuable adaptation. It is another example of how plants gather information about their surroundings and make what might be called strategic decisions about where to invest their growth. Plants can feel vibrations caused by chewing insects. Research has revealed that plants can detect the specific vibrations produced by caterpillars chewing on their leaves and mount defensive responses accordingly. In studies with Arabidopsis plants, recordings of caterpillar feeding vibrations were played back to plants, and those exposed to the sounds produced higher levels of defensive chemicals when later attacked. Critically, the plants did not respond in the same way to other sounds such as wind or the vibrations of non-feeding insects, indicating they can distinguish between threatening and non-threatening vibrational patterns. The mechanism appears to involve specialized mechanoreceptors that convert mechanical vibrations into biochemical signals within the leaf tissue. This vibration sensing adds another dimension to plant perception, allowing them to detect and prepare for threats that are not yet in direct contact with sensitive tissues. It is a form of early warning system that operates through the same medium predators and prey use in the animal world: sound and vibration. The silent response to insect chewing hints at hidden channels of communication and awareness in the plant kingdom. The Venus flytrap counts the number of times its hairs are triggered. The Venus flytrap is famous for its snap trap mechanism, but what is less well known is that it employs a simple counting system to prevent wasting energy on false alarms. Each trap contains trigger hairs, and the trap only closes when two hairs are touched within roughly 20 seconds of each other. This two-touch requirement helps ensure that the object inside is likely to be prey, rather than a grain of dust or raindrop. Even after trapping something, the plant continues to count additional touches before committing to the energy-intensive process of digestion. If the trap receives enough continued stimulation to confirm prey is present, it seals tightly and secretes digestive enzymes. If not, it may reopen and release the object unharmed. This conditional behavior demonstrates a form of decision-making based on accumulated information over time. The Venus flytrap's counting ability is a stark reminder that sophisticated information processing can occur without neurons or a brain. Plants use electrical signals to communicate between different parts. Long before scientists discovered neural networks in animals, plants had evolved their own systems for transmitting electrical signals across their bodies. When a leaf is damaged, electrical impulses can propagate to other parts of the plant within seconds, alerting distant tissues to mount defensive responses. These signals, called action potentials and variation potentials, travel through the vascular system and can be measured with sensitive electrodes. The speed and coordination of these signals allow plants to respond to threats in a unified, organism-wide manner rather than as a collection of independent parts. In some species, electrical signaling is involved in rapid movements, such as the snapping shut of a Venus flytrap or the closing of leaflets in the sensitive plant. The molecular machinery underlying plant electrical signals shares some similarities with animal neurons, including the involvement of ion channels and changes in membrane potential. While not organized into anything like a brain, these electrical communication networks provide plants with a rapid, flexible system of internal coordination. Some plants release warning chemicals when attacked by herbivores. When plants are attacked by insects or other herbivores, many release volatile organic compounds into the air as part of their defensive response. These airborne chemicals, often perceived by humans as the fresh green smell of cut grass or damaged leaves, serve multiple functions. They can directly repel herbivores, attract predators or parasites of the attacking insects, or trigger defensive responses in the plant's own undamaged tissues. Different compounds may be released depending on the identity of the attacker, with some plants producing specific blends that target particular herbivore species. The production and release of these volatiles require metabolic investment, so plants typically regulate their emission according to the severity and type of threat. This chemical signaling system represents an outward-facing aspect of plant intelligence, an ability to project information into the surrounding environment. The scents we associate with lush vegetation are often the echoes of ongoing battles between plants and the creatures that would consume them. Neighboring plants can detect these airborne chemical warnings. The volatile compounds released by attacked plants do not simply drift away unnoticed. Neighboring plants can detect them and respond by preparing their own defenses. This phenomenon, sometimes called talking trees or plant communication, was first documented in the 1980s and has since been confirmed in many species. When undamaged plants receive these chemical signals, they may prime their defensive systems, producing enzymes and compounds that will allow a faster and stronger response if they too are attacked. This priming does not always require direct damage to the receiving plant. Mere exposure to the warning volatiles can shift its internal state toward heightened readiness. The ecological significance of this communication is profound, as it means that an attack on one individual can benefit the defensive preparedness of entire neighborhoods. Critics debate whether plants release these signals to help neighbors, or whether neighbors are simply eavesdropping. But the result is the same. Information flows through the forest air. The green world is far more connected and communicative than it might appear. Trees can share nutrients through underground fungal networks. Beneath the forest floor lies a vast, hidden web of fungal filaments connecting the roots of trees and other plants. These mycorrhizal networks form mutually beneficial relationships, in which fungi receive sugars from plants in exchange for providing increased access to water and mineral nutrients. But the network also allows for the transfer of resources between plants, meaning that nutrients can flow from one tree to another through the fungal pathways. Studies have shown that carbon, nitrogen, phosphorus, and water can all move through these networks, sometimes from well-lit trees to shaded ones that would otherwise struggle to survive. The implications are remarkable. Trees may be supporting each other through shared resources, creating forests that function more like cooperative communities than collections of competing individuals. This underground economy challenges the notion that nature is purely competitive and highlights the profound interdependence of forest ecosystems. The next time you walk among trees, remember that beneath your feet, resources and perhaps even information are quietly flowing. These fungal connections are sometimes called the wood wide web. The intricate mycorrhizal networks linking forest trees have been affectionately nicknamed the wood wide web, evoking the image of an underground internet through which plants share resources and information. This analogy captures the distributed, multi-node nature of the network, where each tree is connected not just to the fungi but, through them, to many other trees. Just as the internet allows information to flow between distant computers, the wood wide web enables biological resources and possibly chemical signals to move between distant plants. The phrase has popularized the idea that forests are not simply collections of individuals, but integrated systems with their own forms of communication and mutual support. While scientists debate the extent to which this network is truly altruistic or simply reflects parasitism and self-interest among the participants, the metaphor has captured the public imagination. It invites us to see forests as living communities, with hidden connections sustaining the visible world above. The wood wide web is a poetic reminder that much of nature's complexity lies beneath the surface. Mother trees can recognize their own seedlings underground. One of the most remarkable findings in forest ecology is that large, established trees, sometimes called mother trees, appear to preferentially send resources through fungal networks to their own genetic offspring. In studies with Douglas fir and other species, researchers found that seedlings growing near their mother trees received more carbon and nutrients via the mycorrhizal network than unrelated seedlings in the same area. This suggests a form of kin recognition operating entirely underground, without any visual or above-ground cues. The mechanism may involve chemical signals in root exudates that allow fungi and parent trees to identify related seedlings. Such preferential treatment could improve the survival chances of offspring, passing the mother tree's genes more successfully to future generations. The existence of this recognition challenges assumptions that plants are incapable of social behavior and reveals a hidden world of family dynamics in the forest. Mother trees nurturing their young through invisible underground channels is one of the most touching images to emerge from modern plant science. Plants may preferentially share resources with genetic relatives. Beyond mother trees supporting their seedlings, a broader pattern has emerged, suggesting that plants in general may favor their kin when distributing resources through shared networks, or even when competing for space. Some studies have found that plants growing alongside relatives produce fewer competitive roots than they would when growing near strangers, a behavior called kin recognition. By reducing competition among relatives, plants may collectively improve the survival of their shared genetic material. This phenomenon has been observed in sea rocket, rice, and several other species, though it is not universal. The mechanisms for kin recognition likely involve chemical signals exchanged through root exudates that allow plants to distinguish related from unrelated neighbors. Such behavior suggests a form of cooperation that natural selection can favor even in the absence of consciousness or intention. The quiet generosity of plants toward their relatives adds another layer of social complexity to the living world beneath and around us. Some plants change their root behavior when near kin versus strangers. When growing in close proximity to other plants, some species adjust their root architecture, depending on whether their neighbors are genetically related or not. Near strangers, plants may produce more extensive, competitive root systems, designed to capture as many resources as possible before the competitor does. Near kin, however, these same plants may restrain their root growth, reducing overlap and potential competition for shared soil resources. This conditional behavior suggests that plants can sense neighbor identity and modulate their growth strategy accordingly. The detection of kin appears to involve chemical cues in root exudates, which differ between related and unrelated individuals. Such behavioral flexibility challenges the idea that plants grow mindlessly according to fixed genetic programs. Instead, it points toward an adaptive responsiveness that takes social context into account. The quiet diplomacy of roots deciding when to compete and when to cooperate unfolds entirely out of sight, beneath the surface of every forest and garden. Plants can adjust their growth based on the identity of neighbors. Beyond distinguishing kin from strangers, plants can also respond to the specific identity of their neighbors and adjust their growth patterns accordingly. Through chemical recognition of root exudates and volatile signals, plants can identify whether they are growing near members of their own species, different species, or even the same individual clone. These identifications influence decisions about how much to invest in roots versus shoots, how to orient branches to avoid shading conflicts, and whether to accelerate flowering and seed production in competitive environments. For instance, some plants will invest more in height growth when surrounded by competitors of the same species, racing toward the light. Others may shift investment to reproduction if they sense they are unlikely to win the competition for space. This ability to tailor growth strategy based on social surroundings demonstrates a form of behavioral plasticity that maximizes survival in complex ecological communities. The green world is more attentive to its neighborhood than we might have imagined. A damaged tree can send warning signals through the fungal network. When a tree is attacked by insects or pathogens, it can transmit warning signals through the mycorrhizal network, alerting neighboring trees to prepare their own defenses. This underground communication allows information about threats to spread beyond the range of airborne volatiles and reach trees that might not otherwise detect the danger until attacked themselves. Experiments have shown that trees connected via fungal networks mount stronger defensive responses than isolated trees when threats are introduced to their neighbors. The signals may be chemical, electrical, or some combination of both, traveling through the fungal filaments that link root systems. This networked early warning system enhances the collective resilience of the forest, allowing communities of trees to respond in concert to shared challenges. The discovery of this phenomenon has deepened our appreciation for forests as cooperative, interdependent systems, rather than mere collections of competing individuals. Even in stillness, trees are exchanging vital information beneath the soil. Plants can anticipate sunrise and prepare their chemistry in advance. Plants possess internal circadian clocks that allow them to anticipate predictable changes in their environment, rather than merely reacting after the fact. One of the most important daily events for a plant is sunrise, when the opportunity for photosynthesis resumes. In the hours before dawn, plants begin activating genes and synthesizing proteins required for efficient photosynthesis, so that by the time sunlight arrives, their molecular machinery is ready to work. This anticipatory preparation gives plants a head start, enabling them to capture light energy more effectively from the very first moments of the day. The same clocks help plants anticipate sunset, preparing for nighttime processes such as the closure of stomata to conserve water. Internal timing is calibrated by light cues, but continues to operate even in constant darkness for several days, revealing a truly endogenous sense of time. The ability to predict and prepare for regular events is a hallmark of adaptive intelligence, quietly at work in every garden and forest. Some plants have internal clocks that run even in total darkness. When plants are placed in constant darkness without any light cues, many continue to display rhythmic behaviors such as leaf movements and gene expression patterns that cycle approximately every 24 hours. This reveals that plants possess endogenous circadian clocks capable of keeping time independent of external signals. The clocks are driven by molecular feedback loops involving specific genes and proteins that rise and fall in concentration throughout the day. These internal timekeepers evolved because the Earth's rotation creates predictable changes in light, temperature, and other conditions, making it advantageous to anticipate rather than simply react. The ability to maintain rhythms in total darkness demonstrates that these clocks are not simply responses to light, but are genuine internal timers. Over time, without external cues, these free-running rhythms may drift slightly, but they remain remarkably consistent. The quiet ticking of plant circadian clocks reminds us that even the most rooted life keeps careful track of time. Plants can predict regular cold stress and activate genes beforehand. In climates where cold nights regularly follow warm days, some plants have evolved the ability to anticipate dropping temperatures and prepare in advance. Using their circadian clocks and temperature sensing, these plants can activate cold resistance genes before temperatures actually fall, producing protective proteins and adjusting membrane composition to prevent freezing damage. This anticipatory response is more effective than waiting until the cold arrives, because molecular adjustments take time to implement fully. By predicting regular patterns of stress, plants can invest their resources more efficiently and minimize damage. The phenomenon demonstrates that plants integrate timing information with environmental sensing to make forward-looking adjustments to their physiology. Just as gardeners cover tender plants before an expected frost, the plants themselves are taking preparatory action. This quiet foresight unfolds nightly in temperate regions around the world, a testament to the adaptive sophistication of plant biology. Climbing plants can anticipate where a support structure will be. Climbing plants face the challenge of locating suitable supports in a complex, three-dimensional environment. Remarkably, some species appear to sense the presence and direction of potential supports before actually touching them, adjusting their growth and circumnutation movements toward these targets. This anticipatory behavior may rely on gradients of light, humidity, or volatile chemicals emanating from nearby structures or other plants. Time-lapse observations of climbing vines reveal purposeful-looking movements that cannot be explained by random growth alone. The ability to predict and reach toward distant supports allows climbers to minimize wasted growth and reach stable anchorage more efficiently. This spatial awareness is especially impressive, given that plants have no centralized visual system. The seeking tendrils of climbing plants demonstrate that purposeful navigation can emerge from distributed sensory processes operating without anything resembling a brain. Some vine tendrils adjust direction before making contact with a pole. Careful time-lapse studies of climbing vines have revealed that their tendrils often change direction and orient toward a nearby pole or support structure before ever making physical contact with it. This suggests that the tendril can sense the presence of the support through some medium other than direct touch, perhaps responding to humidity, reflected light, or chemical cues from the surface. Once the tendril nears the support, its rate of coiling and circling may increase, as if homing in on a target. After contact is made, the tendril coils tightly around the support, often within minutes, anchoring the plant securely. The combination of distance sensing and rapid physical response allows climbing plants to exploit their environment with impressive efficiency. This targeted seeking behavior challenges assumptions that plant movements are purely random tropisms. It hints at a more active form of environmental engagement than we usually attribute to the botanical world. Plants form associations between unrelated stimuli like breeze and light. Associative learning, the ability to link two previously unrelated stimuli and respond to one as a predictor of the other, was long considered a hallmark of animal cognition. Yet recent experiments have suggested that plants can also form such associations. In one study, pea plants learned to associate a breeze with the arrival of light, growing toward the direction of the breeze alone if the two had been reliably paired during training. This behavior suggests that plants can extract predictive relationships from their environment and use them to optimize future responses. The mechanisms underlying this learning remain under investigation, but likely involve chemical and molecular memory systems rather than neurons. The discovery that plants can form associative links between stimuli challenges the boundaries of cognition and raises fascinating questions about the evolution of learning. Even without brains, plants appear capable of building internal models of environmental regularities to guide their behavior. This associative learning was once thought to require a nervous system. For much of the history of psychology and neuroscience, the ability to form associations between stimuli was considered an exclusive property of organisms with nervous systems. From Pavlov's dogs to operant conditioning in laboratory animals, learning was synonymous with neural activity. The discovery that plants can also form such associations has therefore challenged long-held assumptions about what kinds of biological systems can learn. Plants lack neurons, synapses, and anything resembling a brain, yet they achieve functionally similar outcomes using chemical signaling and cellular memory. This suggests that the capacity for learning may not depend on any particular substrate, but rather on organizing principles that can be implemented in various biological architectures. The philosophical implications are profound, forcing us to reconsider what we mean by learning and whether it truly requires consciousness or subjective experience. The humble pea plant's ability to learn is a quiet revolution in our understanding of life and mind. Plants store environmental memories in their cellular chemistry. When plants experience stress, they often undergo chemical and epigenetic changes that persist long after the immediate threat has passed. These changes can alter gene expression, hormone levels, and metabolic pathways in ways that prime the plant for future encounters with similar stresses. In this sense, plants store memories of past experiences within their cellular chemistry, creating a biological record that influences future behavior. A plant that has survived drought, for instance, may be more quickly and effectively primed to respond to subsequent dry spells. These memories are not encoded in neurons, but in the molecular states of cells, including modifications to DNA and associated proteins. The duration of such memories can vary from days to the entire lifespan of the plant, and some can even be transmitted to offspring. This internal record-keeping allows plants to learn from experience and improve their chances of survival in fluctuating environments. Repeated drought can change how a plant responds in the future. Plants that have experienced drought often develop a form of stress memory that alters their physiology and responses to subsequent dry periods. After recovery from an initial drought, these plants may respond more quickly and effectively to a second drought, closing their stomata sooner, adjusting osmotic balance more rapidly, or redirecting root growth toward deeper water sources. This priming effect is thought to result from epigenetic modifications and changes in hormone signaling that persist after the original stress has passed. The phenomenon has been observed in a range of species and has important implications for agriculture, as pre-stressed plants may be more resilient in arid conditions. The ability to learn from past droughts and prepare for future ones demonstrates a form of experiential adaptation that enhances survival. It is a quiet form of wisdom stored in leaf and root, allowing plants to face the uncertainties of climate with greater readiness. These stress memories can be passed to the next generation of plants. Perhaps even more remarkable than individual stress memory is the fact that some plants can transmit this learned resilience to their offspring. Through epigenetic mechanisms, modifications to the expression of genes without changing the underlying DNA sequence itself, stress-related information can be inherited by seeds and seedlings. The progeny of drought-stressed plants may, for example, display improved drought tolerance even if they themselves have never experienced water shortage. This transgenerational inheritance allows environmental lessons to be passed down, potentially improving the survival chances of the next generation in similar conditions. The phenomenon has been documented in Arabidopsis and other species, though its prevalence across the plant kingdom is still being studied. Transmission of stress memories is a form of biological information transfer that operates without any instruction from parent to offspring. It is as if the parent plant encodes a whispered warning into its seeds, preparing them for challenges it has already faced. Epigenetic changes allow plants to inherit survival information. Epigenetics refers to modifications that affect gene activity without altering the DNA sequence itself, such as DNA methylation and histone modifications. In plants, epigenetic changes can be induced by environmental stresses and may persist across cell divisions, influencing how genes are expressed for the remainder of the plant's life. Even more remarkably, some of these modifications can be passed through seeds to future generations, allowing offspring to inherit not just genes but also information about the environments their parents experienced. This creates a form of Lamarckian inheritance, where acquired traits can be transmitted to descendants, a concept long dismissed in animal biology but increasingly recognized in the plant world. Epigenetic inheritance allows plants to adapt rapidly to changing environments without waiting for genetic mutation and selection. This inherited environmental intelligence adds a transgenerational dimension to plant learning and resilience. Plant roots explore soil, like an organism searching for food. The way roots grow through soil is not a passive process, but an active exploration, resembling the foraging behavior of animals seeking food. Root tips constantly sense their surroundings, adjusting direction and growth rate based on the distribution of water, nutrient gradients, obstacles, and even the presence of neighboring roots. When a root encounters a pocket of fertile soil, it may branch extensively to exploit the resource. When it meets a barrier, it may redirect or pause and try a new path. This exploratory behavior involves real-time integration of multiple sensory inputs and continuous adjustment of growth strategy. Darwin likened root tips to the brains of lower animals, and modern research supports the idea that roots display purposeful, flexible behavior. The analogy to foraging is not merely metaphorical. Roots make trade-off decisions about where to invest limited resources in search of essential supplies. Beneath the ground, a complex search is always underway. Root tips contain densely packed sensory cells. The very tip of a plant root is one of the most remarkable structures in biology, densely packed with sensory cells that gather information about the surrounding soil environment. These cells can detect gravity, touch, moisture, chemical gradients, and even the electrical signals of nearby roots. The root cap protects the dividing cells of the root meristem and also contains statocytes that sense gravity, enabling the root to orient downward. Just behind the cap lies the transition zone, where incoming sensory information is integrated and translated into growth responses. The concentration of sensory function in the root tip allows for precise, localized decision-making about which direction to grow next. No other part of the plant is so densely devoted to environment sensing and response coordination. Darwin's comparison of the root tip to a brain was remarkably prescient, capturing the functional importance of this tiny organ in guiding the plant's underground journey. Darwin compared root tips to the brains of lower animals. In his later years, Charles Darwin became fascinated by plant movement and conducted extensive experiments with his son Francis. Together they published The Power of Movement in Plants in 1880, in which Darwin made a remarkable comparison. He likened the root tip to the brain of a lower animal, directing movements of the adjoining parts. This analogy was controversial at the time and remains so today, but it captured the idea that the root tip is a site of sensory integration and behavioral control. Modern research has largely supported Darwin's intuition, showing that root tips contain specialized zones where signals are processed and growth decisions are made. While plants lack neurons and brains, the functional parallels are striking. Roots sense, integrate and respond in ways that parallel animal behavior, just on different timescales and with different machinery. Darwin's observation invites us to see intelligence not as the exclusive property of animals, but as a feature of adaptive living systems more broadly. Roots can pause, reverse, and try new directions when blocked. When a growing root encounters an obstacle such as a stone or compacted soil, it does not simply stop. Instead, it may pause, probe the barrier, and eventually change direction to find a way around. This adaptive behavior involves sensing the physical impediment, re-evaluating the local environment, and redirecting growth resources to cells on the side that offers a more promising path. In some cases, roots have been observed to reverse briefly before attempting a new direction, as if reassessing their options. This flexible response to obstacles allows roots to navigate complex underground environments and reach vital resources despite physical barriers. The analogy to problem solving is hard to resist, even though the process operates through biochemical signaling rather than conscious deliberation. The root's ability to overcome obstacles one step at a time reveals a quiet persistence that underpins plant success in unpredictable soils. The transition zone in roots shows neuron-like electrical activity. Just behind the root tip is a region known as the transition zone, where cells have completed division but not yet begun significant elongation. This narrow band of tissue displays intense electrical activity, including action potential-like signals and oscillating ion fluxes that resemble some aspects of neural signaling in animals. Some researchers have proposed that this zone functions as a kind of command center, integrating sensory information from the root cap and coordinating growth responses throughout the root. The analogy to neurons is not perfect, as plant cells lack synapses and the molecular machinery differs in many ways, but the functional parallels are intriguing. The transition zone may be where the plant decides, in some sense, which direction to grow next. The discovery of neuron-like activity in root tips has fueled debate about whether plants possess a form of cognition and has inspired new approaches in the emerging field of plant neurobiology. Roots prefer nutrient-rich paths and avoid dead ends. As roots navigate the soil, they display preferences that suggest they are evaluating the quality of different paths. Roots tend to grow toward patches of soil rich in nitrogen, phosphorus, and other essential nutrients, branching extensively when they encounter such pockets. Conversely, they tend to avoid areas where resources have been depleted or where toxic substances are present. In maze-like experimental setups, roots have been observed to avoid dead ends and choose routes that lead to resources more reliably. While we should be cautious about attributing intention to these behaviors, the functional outcome is the same as if the root were making choices about where to invest its limited growth energy. This resource-seeking behavior is critical for the survival and competitiveness of plants in heterogeneous soils. The navigational skill of roots underscores how effectively plants solve spatial problems without any centralized nervous system. Two genetically identical plants may choose different root paths. Even when two plants have the exact same genetic makeup and are grown in identical conditions, their root systems often develop differently, taking divergent paths through the soil. This variability arises because the root growth process is not entirely deterministic. Small fluctuations in local conditions, signaling molecules, and growth dynamics introduce randomness into the trajectory each root takes. This behavioral variability may be adaptive, spreading the risk that all roots will encounter the same obstacle or dead end simultaneously. It is a form of bet hedging, where the plant genetically programs for a range of possible outcomes rather than a single fixed strategy. The result is that siblings or clones can explore different niches and improve the collective chance that at least some roots will find essential resources. This stochastic element in root growth adds yet another layer of complexity to plant behavior and challenges the notion that plants are merely mechanical in their responses. This variability may be a survival strategy called bet hedging. Bet hedging is an evolutionary strategy in which organisms produce variable offspring or behaviors to reduce the risk of catastrophic failure in unpredictable environments. In plants, variability in root paths, germination timing, and seed size all serve this function, ensuring that not all individuals will fail simultaneously if conditions turn unfavorable. By introducing randomness into their development, plants spread risk across multiple outcomes, improving long-term survival of the population. This strategy is especially important in environments where conditions fluctuate unpredictably, such as deserts where rainfall is erratic, or forest floors where light gaps appear randomly. Bet hedging is a form of population-level intelligence, an evolved wisdom that accepts uncertainty and plans for multiple futures. The quiet variability in how individual plants develop is evidence of deep evolutionary foresight encoded at the genetic level. It is a reminder that intelligence can be expressed not only in individual behavior, but also in the distribution of behaviors across a population. plants can balance their internal resources between roots and shoots. Plants face a fundamental allocation problem: how to divide limited resources between growth above ground to capture light and growth below ground to acquire water and nutrients. This trade-off is constantly adjusted based on environmental conditions. When a plant experiences drought, it may shift resources toward root development to seek out deeper water sources. When shaded by competitors, it may invest more in shoots and leaves to reach the sunlight. This dynamic rebalancing requires integrating information from all parts of the organism and making what amounts to a strategic decision about where to invest. Hormones such as auxin and cytokinin mediate these allocation shifts, flowing through vascular tissues to coordinate whole plant responses. The ability to balance growth investments demonstrates an organism-level form of resource management that maximizes the chance of survival and reproduction. The plant is constantly asking in its chemical language where the greatest return on investment lies. Plants weigh competing signals and make trade-off decisions. In their complex environments, plants constantly receive multiple, sometimes conflicting signals about where to grow and how to allocate resources. A root might detect both a source of nutrients and a zone of harmful salinity in the same direction. A stem might sense light coming from one direction and wind stress from another. In these situations, plants do not simply respond to the strongest signal, but appear to weigh competing inputs and make integrated decisions that balance costs and benefits. This decision-making involves the integration of multiple sensory pathways at the cellular and whole plant level, often mediated by hormones and signaling cascades. The result is often a compromise, a growth direction or allocation pattern that optimizes overall survival, even if it does not perfectly satisfy any single criterion. This ability to weigh trade-offs is a hallmark of adaptive behavior and demonstrates a kind of practical wisdom embedded in plant physiology. The quiet calculations occurring in every stem and root are more sophisticated than they might first appear. Some plants grow towards salty soil if nutrients outweigh the risk. Salt stress is normally harmful to plants, damaging cells and disrupting water uptake. Yet some roots will grow toward moderately salty soil if that zone also contains high concentrations of essential nutrients. This behavior demonstrates that plants are capable of cost-benefit analysis, weighing the danger of salt against the reward of nutrients and adjusting growth accordingly. When nutrients are scarce elsewhere, the potential gain may outweigh the risk, prompting roots to venture into less than ideal territory. If the balance shifts, with salt concentrations rising or nutrient availability falling, the same plant may redirect its roots away from the risky zone. Such flexible, contextual decision-making is difficult to explain without invoking some form of evaluation and integration of multiple environmental cues. The willingness to accept a trade-off in pursuit of overall benefit is another example of plant intelligence operating beneath the surface. It is a gamble that roots make every day, invisible to the world above. Plants can distinguish mechanical damage from insect feeding. When a leaf is damaged, it matters greatly whether the injury was caused by a falling branch, hail, or an insect's mandibles. Plants have evolved the ability to discriminate between these types of damage and mount appropriate responses. Mechanical damage from hail or wind triggers wound-healing responses, but it may not activate the full suite of anti-herbivore defenses. Insect feeding, in contrast, is often accompanied by saliva or other chemical cues that the plant can detect, triggering production of defensive compounds targeted specifically at the attacker. Some plants can even differentiate between specialist and generalist herbivores and tailor their defenses accordingly. This discrimination prevents the plant from wasting resources on inappropriate responses and allows it to fine-tune its defense strategy. The ability to read the signature of an attacker from the nature of a wound is a sophisticated form of contextual sensing that reveals deep evolutionary refinement. They tailor defensive chemicals to match the type of attacker. Plants do not rely on a single fixed defensive strategy, but instead produce an array of chemical compounds that can be adjusted depending on the identity of the current threat. When attacked by caterpillars, a plant may produce one set of toxins or digestibility-reducing compounds. When attacked by aphids, it may switch to an entirely different suite of chemicals. This targeted response is triggered by specific cues in the attacker's saliva or feeding behavior, allowing the plant to customize its defense in real time. The result is more effective protection with less wasted resources, as the plant does not invest in defenses that would be ineffective against the current herbivore. This tailored chemical warfare demonstrates that plants are continuously evaluating their enemies and adjusting their response profiles. The sophistication of these defenses rivals the immune systems of many animals, even though the underlying mechanisms are entirely different. Each attack is met with a bespoke chemical counteroffensive. Some plants attract predators of the insects eating them. In one of nature's most elegant defensive strategies, some plants respond to herbivore attack by releasing volatile chemicals that attract the natural enemies of their attackers. When caterpillars begin feeding on maize, for example, the damaged leaves release a bouquet of compounds that lures parasitic wasps, which lay their eggs inside the caterpillars and kill them. This indirect defense effectively recruits an army of bodyguards from the surrounding environment, reducing herbivore pressure without the plant having to produce expensive toxins. The chemicals released are specific enough to attract predators or parasitoids suited to the current pest, demonstrating remarkable precision in the plant's call for help. This form of interspecies communication blurs the line between isolated organism and participant in a broad ecological network. The plant's distress signal travels through the air and reshapes the behavior of insects it has never directly encountered. It is a silent cry for help that echoes across the ecosystem, answered by tiny but deadly allies. Damaged leaves can trigger defenses in undamaged leaves far away. When one leaf of a plant is attacked or wounded, signals can travel through the plant's vascular system and trigger defensive responses in distant, undamaged leaves. This systemic acquired resistance allows the entire plant to prepare for further attack, even before herbivores or pathogens have spread from the initial site of damage. The signals involved include electrical impulses, hormonal waves, and mobile peptides that travel rapidly through phloem and xylem. Within minutes to hours, undamaged leaves may begin producing defensive compounds, thickening cell walls, or synthesizing enzymes that deter feeding. This whole plant coordination is analogous to an immune response in animals, where a local infection triggers systemic alertness. The ability to broadcast threat information internally allows plants to mount anticipatory defenses rather than waiting for each leaf to be attacked in turn. It is a form of organism-level awareness and preparedness that improves survival in hostile environments. Calcium waves carry injury signals through plant tissues. One of the key messengers that transmit injury signals from damaged to undamaged parts of a plant is the calcium ion. When a cell is wounded, local increases in calcium concentration trigger cascading waves that propagate through adjacent cells at remarkably high speeds. These calcium waves can travel through both the vascular system and direct cell-to-cell connections, reaching distant tissues within seconds to minutes. Once the wave arrives at a target cell, it triggers downstream signaling events that prepare the tissue for defense. Calcium signaling is fundamental to many aspects of plant biology, from growth regulation to stress responses, making it a versatile and rapid means of internal communication. The propagation of calcium waves is one of the closest analogues to neural transmission that plants possess, allowing for real-time coordination across the organism. This ionic language pulses silently through every stressed plant, coordinating its response to injury. Plant hormones coordinate responses across the entire organism. Plants rely on a suite of hormones, including auxin, cytokinin, ethylene, gibberellins, abscisic acid and jasmonic acid, to coordinate growth and responses across their entire body. These chemical messengers are produced in specific tissues and travel to distant sites, where they regulate gene expression, cell division, and metabolic activity. Together, they form an integrated communication network that allows the plant to function as a unified organism, rather than a collection of independent parts. When drought strikes, abscisic acid rises and promotes stomatal closure to conserve water. When a herbivore attacks, jasmonic acid triggers defensive pathways throughout the plant. The balance and interplay of hormones enable plants to make trade-offs between growth, defense, and reproduction, depending on current conditions. This hormonal symphony plays out continuously, tuning the plant's behavior to the ever-changing environment. It is the chemical equivalent of a nervous system, coordinating the silent decisions that shape every aspect of plant life. Auxin flows through plants like signals through a nervous system. Auxin is one of the most important plant hormones, involved in regulating cell elongation, root initiation, vascular development, and responses to light and gravity. It is transported directionally through plant tissues by specialized carrier proteins, creating gradients that guide growth and shape the plant's architecture. This polarized transport allows auxin to convey positional information, telling cells where they are in the organism and how they should behave. The flow of auxin through the plant has been compared to the flow of signals through a nervous system, carrying information that influences distant parts of the body. When a stem bends toward light or a root grows downward, auxin redistribution is underlying the response. Disruptions in auxin transport lead to dramatic alterations in plant form, underscoring its central role in coordinating development. The analogy to neural signaling is imperfect, but it captures the essential idea that auxin is a carrier of information across space and time within the plant. Plants can modulate hormone levels based on environmental context. Plants do not simply produce fixed amounts of each hormone, but dynamically adjust their levels in response to changing conditions. When water becomes scarce, abscisic acid rises. When shade intensifies, auxin and ethylene are modulated to drive rapid elongation toward light. These adjustments are not hardwired reflexes, but emerge from complex regulatory networks that integrate multiple environmental inputs. The ability to modulate hormone levels allows plants to optimize their physiology and behavior for the current situation, whether that means investing in growth, defense, reproduction, or dormancy. This hormonal flexibility is akin to the way animals adjust their neuroendocrine systems in response to stress or opportunity. The result is a highly adaptable organism that can respond appropriately to a vast range of circumstances. Each plant's hormonal profile is a dynamic snapshot of its current needs and challenges, constantly recalibrated as conditions shift. Some plants eavesdrop on the chemical warnings of other species. Plants do not only detect warning signals from members of their own species, some can eavesdrop on the volatile chemical cues released by entirely different plant species growing nearby. When a neighboring plant of another species is attacked and releases defensive volatiles, a sensitive listener may detect these chemicals and prepare its own defenses in advance. This cross-species communication means that ecological communities can share threat information across taxonomic boundaries, potentially improving collective resilience. The ability to eavesdrop suggests that plants are attuned to a broad chemical vocabulary of alarm signals, not just the specific compounds produced by their own kind. It is an example of how interconnected the natural world truly is, with information flowing between organisms that never physically touch. The forest air carries messages from many speakers, and those who can interpret them gain an advantage. This chemical eavesdropping is another reminder that plants are active participants in a complex web of ecological communication. A damaged tomato can unintentionally warn a nearby bean plant. In experiments demonstrating cross-species chemical communication, researchers found that when tomato plants were damaged by herbivores, nearby bean plants responded by upregulating their own defensive pathways. The tomatoes were not intentionally sending signals to the beans, they were simply releasing volatiles as part of their own defense. But the beans, growing in close proximity, detected these airborne cues and interpreted them as warnings to prepare for possible attack. This incidental communication illustrates how plants can benefit from the misfortunes of their neighbors, even unrelated ones, by remaining alert to chemical changes in the air. The phenomenon has practical implications for agriculture, suggesting that intercropping certain species might enhance overall pest resistance. It also highlights the pervasive role of volatile signaling in shaping plant communities. The silent conversation between tomatoes and beans is just one example of the many hidden dialogues occurring in every garden and field. Plants in forests share a chemical vocabulary of danger signals. Across a forest, many different species of plants release volatile organic compounds when stressed or damaged, and over evolutionary time, certain signals have become commonly recognized across wide ranges of species. This shared chemical vocabulary allows plants to communicate about threats in a kind of universal language, at least within their local ecological context. The most common danger signals include compounds like methyl jasmonate and green leaf volatiles, which many plants both produce and respond to. This convergence likely evolved because detecting any sign of danger, regardless of source, confers a survival advantage. The existence of a shared signaling system transforms the forest from a collection of isolated individuals into a communicative network, where information flows and defenses can be coordinated at scale. Even as you walk through a quiet woodland, chemical conversations are constantly taking place in the air around you. The botanical world speaks a language we are only beginning to understand. Trees can allocate more carbon to struggling neighbors. Through the mycorrhizal fungal networks connecting their roots, trees can transfer carbon, in the form of sugars produced by photosynthesis, to neighboring trees that are struggling. This resource sharing means that a shaded tree receiving insufficient light for its own photosynthesis may be supported by its better-lit neighbors. Studies tracking isotopically labeled carbon have shown that sugars can move from one tree to another across the network, sometimes favoring those most in need. Whether this transfer represents active sharing or passive leakage is debated, but the functional outcome is clear. The network allows resources to flow towards scarcity. This carbon support may help maintain forest stability by preventing individual trees from dying and creating gaps that would expose others to wind or pests. The willingness of well-off trees to support their neighbors suggests a level of ecological cooperation that challenges competitive models of nature. The forest is not merely a battleground, but also, in part, a community of mutual aid. Dying trees sometimes transfer nutrients to younger ones. When a large, mature tree is dying, it sometimes appears to transfer its remaining nutrients to younger trees in its vicinity through the fungal network. This end-of-life resource dump may help ensure that the legacy of the dying tree persists in the survival of its neighbors or offspring. Some researchers have observed spikes in nutrient transfer from old trees to young ones in the months before death, suggesting an active process rather than mere decay. Whether this represents intentional generosity or simply a consequence of failing root function is uncertain, but the effect is the same. Resources are recycled within the forest community. Such transfers may be especially important in old-growth forests, where mature trees possess vast nutrient reserves accumulated over centuries. The dying tree's final act may be one of giving, enriching the soil and the next generation. It is a poignant image, the forest elder passing its wealth to the young. Some fungal networks appear to broker trades between different trees. The fungi that form mycorrhizal networks with tree roots are not passive conduits, but active participants in determining how resources flow. Some evidence suggests that fungi may preferentially channel resources to trees that provide the best return in sugars, effectively brokering trades and incentivizing generosity. This means the network may operate somewhat like a marketplace, where resources move toward those who can pay the highest prices. Fungi benefit from the overall health of their host trees, so they may have evolved strategies to maximize network productivity. The economics of the Wood Wide Web are complex and not yet fully understood, but the idea that fungi actively manage resource flows adds another layer of intelligent coordination to forest ecology. The forest floor may host a hidden economy of exchanges, with fungi as the brokers ensuring the system functions smoothly. It is a reminder that cooperation and trade occur at scales and in forms we rarely perceive. Parasitic plants can sniff out their preferred hosts. Some parasitic plants, which survive by tapping into the vascular systems of other plants to steal water and nutrients, are remarkably adept at locating suitable hosts. They can detect volatile chemicals emitted by nearby plants and grow preferentially toward the species that will provide the best resources. Studies have shown that parasitic plants like dodder can distinguish between more and less nutritious host species based on chemical cues alone, before making physical contact. This olfactory-like sensing allows them to invest their limited growth energy wisely, avoiding hosts that would offer poor returns. The specificity of host detection is often remarkable, with some parasites rejecting multiple potential hosts in favor of a preferred target. This targeted seeking behavior is a sophisticated adaptation that maximizes the parasite's chances of success. Even in the shadowy world of parasitism, there is a form of intelligence at work, guiding growth toward opportunity. Dodder vines grow toward nutritious hosts before making contact. Dodder is a parasitic vine that lacks the ability to photosynthesize as an adult and must attach to host plants to survive. Remarkably, dodder seedlings can detect volatile compounds released by nearby plants and grow toward the most nutritious options even before touching them. In experiments, dodder consistently preferred wheat and tomato over less nutritious hosts when given a choice, orienting its growth toward the chemical signatures of these preferred targets. Once it reaches a suitable host, the dodder coils around the stem and penetrates the tissue with specialized structures called haustoria. The vine's ability to evaluate host quality at a distance and adjust its growth accordingly demonstrates a highly refined sensory system. Dodder's seeking behavior is a vivid illustration of how parasitism can evolve alongside sophisticated perceptual abilities. The plant is, in effect, hunting for its prey, guided by invisible scent trails. Parasites adjust their feeding strategy based on host condition. Once attached to a host, parasitic plants often modulate how aggressively they extract resources depending on the condition of the host. If the host is healthy and productive, the parasite may draw heavily. If the host becomes stressed or weakened, some parasites will reduce their extraction to avoid killing the host prematurely. This strategic restraint makes sense from an evolutionary perspective. A parasite that kills its host too quickly loses its source of sustenance. Some parasites even appear to calibrate their feeding rates to match host photosynthesis, taking only what the host can spare without suffering severe harm. This conditional behavior suggests a form of dynamic interaction between parasite and host, with feedback mechanisms adjusting the flow of resources. The relationship is exploitative, but not without a certain dark wisdom about sustainable extraction. Even parasites must balance short-term gain against long-term survival. Some plants change leaf shape to mimic the plant they climb. Mimicry is usually associated with animals, but at least one remarkable plant, the Bokila vine of the South American temperate rainforest, appears to mimic the leaves of the trees it climbs. On a single vine, leaves can take on different shapes, colors, and sizes, depending on which host tree the vine is attached to. When the same vine moves to a different host species, its new leaves may resemble those of the new host rather than the old. This adaptive mimicry is thought to protect the vine from herbivores that search for specific leaf shapes. How the plant perceives and replicates the form of its host's leaves is not fully understood and remains the subject of active research. Proposed mechanisms include detection of airborne chemicals, gene transfer from host to vine, or even responses to light patterns reflected from the host. The Bokila vine's mimicry is one of the most astonishing examples of apparent intelligence in the plant world. The Bokila vine can mimic multiple host species with different leaves. What makes the Bokila vine especially remarkable is its ability to mimic not just one but multiple host species, sometimes on the same vine at the same time. As the vine grows and encounters different trees, its leaves shift in appearance to resemble each successive host, producing a patchwork of leaf forms that would seem impossible without sophisticated sensing and control mechanisms. This plasticity implies that the vine continually monitors its surroundings and adjusts its developmental program accordingly. The precision of the mimicry, matching not only leaf shape but also vein patterns and coloration, is extraordinary. Researchers remain uncertain how the vine accomplishes this, with hypotheses ranging from volatile chemical detection to horizontal gene transfer. Whatever the mechanism, Bokila challenges our understanding of what plants can perceive and how they can respond. It is a living demonstration that the plant world holds surprises far beyond our current imagination. This mimicry may be guided by airborne chemical or light cues. The mechanism by which the Bokila vine achieves its remarkable leaf mimicry remains one of the most intriguing puzzles in plant biology. Some researchers suggest that the vine may detect volatile chemicals released by its host and use these as templates to guide leaf development. Others propose that the vine might sense the spectrum of light reflected from nearby leaves and adjust pigmentation and shape accordingly. A more speculative hypothesis involves horizontal gene transfer, where genes from the host plant could be incorporated into the vine's genome. As of yet, no single mechanism has been definitively proven, and the answer may involve a combination of sensory modalities. Regardless of how it works, the phenomenon forces us to reconsider the limits of plant perception and phenotypic plasticity. The Bokila vine invites wonder and reminds us that the natural world still holds secrets waiting to be discovered. Carnivorous plants use sophisticated traps to catch prey. Carnivorous plants have evolved an astonishing array of trapping mechanisms to capture and digest animals, typically insects. Snap traps like those of the Venus flytrap close in fractions of a second, triggered by sensitive hairs. Pitcher plants lure prey with nectar and attractive colors, then trap them in slippery pitchers filled with digestive fluids. Sundews use sticky droplets on tentacle-like leaves to ensnare insects, then slowly fold the leaf around the victim. Bladderworts generate vacuum traps that suck in prey at speeds nearly too fast to see. Each of these mechanisms requires precise coordination of sensing, triggering, and digestion, all without a central nervous system. The engineering elegance of carnivorous traps has inspired biomimetic research and leaves no doubt that plants can solve complex survival problems. The Venus flytrap only closes when two hairs are touched within seconds. The Venus flytrap's remarkable trap mechanism incorporates a counting and timing system that prevents wasted energy. Each trap lobe contains 3 to 5 small sensory hairs, and the trap will only snap shut if at least 2 hairs are triggered within approximately 20 seconds. A single touch is not enough, which helps the plant avoid closing its trap on inedible debris like raindrops or falling leaves. This threshold system functions remarkably like a neural circuit, comparing stimulus events across time. The trigger hairs generate electrical signals, action potentials, that are summed by the plant's tissues before the trap is activated. If the insect continues to struggle after the trap closes, additional touches accelerate the release of digestive enzymes. The Venus flytrap's behavior is a compelling example of computation-like processes operating within a plant's simple tissues. This counting mechanism prevents wasted energy on false alarms. The Venus flytrap's requirement for multiple touches before closing represents an elegant solution to a cost-benefit problem. Closing and reopening the trap consumes significant energy and can only be repeated a limited number of times in each trap's lifespan. If the trap closed on every random stimulus, the plant would exhaust its resources without catching enough prey to compensate. By requiring 2 stimuli in quick succession, the trap dramatically increases the probability that a genuine insect moving around inside the trap is present. This threshold also provides a simple form of discrimination, allowing the plant to filter noise from signal. After trapping prey, the plant continues to count further touches, using the intensity of struggle as an indicator of prey size and nutritional value. The flytrap's behavior illustrates how plants can evolve sophisticated decision rules to optimize energy expenditure in uncertain environments. The bladderwort sucks prey into its trap in less than a millisecond. The bladderwort is an aquatic carnivorous plant whose trapping mechanism ranks among the fastest movements in the entire plant kingdom. Its bladder-shaped traps are maintained under negative pressure, and when a tiny invertebrate brushes against trigger hairs near the entrance, the trap door opens and water rushes in, carrying the prey with it. This entire process, from trigger to capture, can occur in less than a millisecond. The speed is achieved through mechanical energy stored in the deformed walls of the trap, a system that requires no muscular action. Once the prey is inside, the trap reseals and digestion begins, with nutrients absorbed directly through the trap walls. The bladderwort's vacuum traps are marvels of hydraulic engineering, evolved to capture microscopic organisms invisible to the naked eye. These tiny plants demonstrate that the race to build faster traps has pushed evolution to the very limits of biomechanical possibility. Carnivorous plants show conditional logic in their responses. Carnivorous plants do not simply react to all stimuli with the same response. They exhibit conditional logic that tailors their actions to the situation. The Venus flytrap, for example, only closes if 2 hairs are touched within a narrow time window, filtering out single accidental touches. Once closed, the plant does not immediately release digestive enzymes, but waits for further stimulation from trapped prey to confirm that something nutritious is inside. If the prey is too small or non-nutritive, the trap may reopen without digesting, saving costly enzyme production for worthwhile meals. Sundews adjust the degree and speed of tentacle bending according to the size and struggles of the prey. These if-then behaviors allow plants to conserve resources while maximizing success, mimicking decision-making processes found in animals. Such conditional responses blur the line between reflexive reaction and adaptive choice. Plants can integrate multiple environmental signals before responding. Rather than responding to a single cue in isolation, plants often integrate information from multiple sources before making a decision. A plant might assess light levels, soil moisture, nutrient availability, temperature, and the presence of neighbors all at once, combining these inputs to determine how to grow or when to flower. This integration allows plants to fine-tune their responses to complex, changing environments, rather than relying on fixed reactions. The process requires comparing signals that operate on different timescales and arise from different tissues, demanding a form of distributed information processing. Hormones, electrical signals, and chemical gradients all contribute to this integration, working in concert, much like parallel circuits in a computer. Such signal integration underlies the remarkable plasticity of plant development and behavior. It demonstrates that plants are sophisticated processors of environmental information, constantly computing the best path forward. This signal integration operates like biological computation. The processes by which plants integrate environmental signals bear a striking resemblance to computation, though the hardware involved is radically different from electronic circuits or nervous systems. Gene regulatory networks serve as logic gates, switching pathways on or off, depending on the combination of inputs. Hormonal gradients can encode continuous values that are compared and contrasted across tissues. Electrical signals provide rapid binary-like communication that coordinates distant parts of the plant in real time. Some researchers now describe plant physiology in terms of Boolean logic, network theory, and decision algorithms that were once reserved for brains and computers. This perspective does not imply that plants think consciously, but it highlights how computation can emerge from biochemical processes. Understanding plants as computing systems opens new avenues for synthetic biology and bio-inspired engineering. Plants use receptors to detect specific chemical compounds. Like animals, plants possess specialized receptor proteins on or within their cells that bind to specific chemicals, triggering downstream responses. These receptors can detect hormones produced internally, volatile compounds from other plants, and chemicals released by herbivores or microbes. When a ligand binds to its receptor, it initiates a cascade of molecular events that alter gene expression, protein activity, or ion fluxes. The specificity of receptors allows plants to distinguish between different signals and mount appropriate responses to each. For example, jasmonic acid receptors detect damage-related hormones, while ethylene receptors respond to ripening signals. This molecular recognition system underpins the precision of plant sensory abilities and behavior. Receptors are the first step in an information processing chain that translates chemical cues into meaningful action. Signal cascades allow plants to amplify and interpret messages. When a receptor detects a signal, it does not directly produce a response. Instead, it triggers a signal transduction cascade that amplifies and modulates the initial message. These cascades involve sequential activation of kinase enzymes, second messengers like calcium ions, and transcription factors that ultimately alter gene expression. The cascade structure allows a small initial signal to produce a large cellular response, and multiple regulatory checkpoints provide opportunities for modulation. Plants can enhance or dampen signals depending on their internal state or other concurrent inputs, achieving remarkably nuanced control. This architecture permits integration of multiple inputs, temporal filtering, and context-dependent output. Signal cascades are the molecular logic that transforms raw perception into coherent behavior. Through cascades, plants translate environmental information into adaptive growth and defense. Plants can produce different defensive compounds for different threats. When faced with herbivores, pathogens, or competing plants, many species can deploy a diverse arsenal of defensive compounds tailored to the specific challenge. Chewing insects may trigger production of protease inhibitors that hinder digestion, while piercing sucking insects induce different toxins that deter feeding. Fungal pathogens can provoke synthesis of antimicrobial phytoalexins, whereas bacterial invasion may stimulate different biochemical defenses. Some plants can even distinguish between closely related herbivore species and adjust their defensive cocktail accordingly. This flexibility requires sophisticated perception and a branched network of biosynthetic pathways that can be selectively activated. The diversity of defenses is thought to reduce the likelihood that any single pest will evolve complete resistance. Chemical warfare in the plant world is remarkably discriminating and endlessly inventive. Some defenses are only activated when multiple conditions are met. Plants often hedge their bets when deciding whether to mount costly defenses, requiring more than one signal before committing. A plant might ignore a single touch but initiate a response if repeated touches occur, or it might require both herbivore damage and the presence of specific chemical elicitors before producing toxins. This conditionality prevents the plant from overreacting to noisy or false signals, while ensuring a robust response when a genuine threat is confirmed. The logic can be understood as AND gates in a biological circuit. Defense is triggered only if condition A and condition B are both satisfied. By layering such conditions, plants achieve fine-grained control over resource allocation and avoid wasting energy on unnecessary defense. This strategy may also reduce the risk that herbivores will evolve resistance, since defense expression is unpredictable. Conditional activation is a hallmark of intelligent, flexible responses in the plant kingdom. This conditional responding resembles logical if-then processing. When a plant only mounts a defense if multiple criteria are met, its behavior can be described using the same formal logic used to model circuits and software. If signal A is present and signal B is present, then produce defensive compound C. This if-then structure is one of the simplest building blocks of computation and is found throughout the plant world. Such logic enables sophisticated decision-making despite the absence of a central nervous system. The molecular machinery that evaluates these conditions includes receptors, kinases, transcription factors, and feedback loops that together function as biological comparators. Researchers have begun to model these pathways using Boolean networks and flowcharts, revealing striking parallels to artificial decision systems. Recognizing conditional logic in plants reframes our understanding of cognition as something not exclusive to brains. Intelligence in this broader sense is the capacity to process information and act adaptively. Plants reconfigure their response thresholds based on past experience. After encountering a particular stress, many plants adjust their sensitivity so that future responses are triggered more quickly or at lower thresholds. This phenomenon, called priming, is a form of memory that prepares the plant for repeated or ongoing challenges. A plant that has suffered a herbivore attack, for example, may require less stimulation to activate its defenses the next time an attacker appears. Priming can persist for days, weeks, or even across growing seasons, and in some cases can be passed to offspring. The molecular basis of priming involves changes in chromatin structure, hormone levels, and signaling protein pools that keep the plant in a ready state. Priming provides an ecological advantage in environments where certain threats are likely to recur. It is a clear demonstration that plants learn from experience and adapt accordingly. This priming allows faster responses to repeated threats. Primed plants do not begin each encounter from scratch. They carry a memory of past threats that allows them to mount defenses more rapidly and sometimes more strongly upon re-exposure. This speed advantage can mean the difference between repelling an herbivore before it causes serious damage and suffering significant tissue loss. Studies have shown that primed plants may activate defense genes within minutes, whereas naive plants require hours. The energy invested in maintaining a primed state is more than compensated for by the improved efficiency of the response when threats recur. Priming also reduces the time lag during which the plant is vulnerable, closing the window of opportunity for attackers. This temporal enhancement of defense illustrates how experience can reshape plant physiology for the better. Priming is evidence that plants live and learn from their histories. Plant roots can distinguish between different soil textures. As roots grow through soil, they can sense and respond to differences in particle size, compaction, and pore structure. In finer or more compacted soils, roots may grow more slowly and invest more in structural support, while in looser, sandier soils, they may extend rapidly in search of water and nutrients. Root tips possess mechanosensitive cells that detect physical resistance and adjust growth direction to navigate around obstacles. The ability to distinguish textures optimizes root architecture for the conditions encountered, maximizing resource acquisition and anchorage. Different genotypes may show varying degrees of this plasticity, an important trait for crop breeding. By sensing and adapting to physical structure, roots effectively feel their way through the underground environment. This tactile exploration is an essential component of plant intelligence hidden beneath our feet. Roots may coordinate growth strategies across split environments. When a single plant's root system spans two contrasting environments, such as one side with ample water and one dry, the plant can coordinate its allocation strategy across both regions. Roots in the favorable zone may proliferate extensively to exploit available resources, while roots in the poor zone may slow their growth or die back. This differential investment is coordinated at the whole plant level, requiring communication between distant root tips and sometimes the shoot. Split root experiments have revealed that plants can weigh options and direct resources toward the most productive sites. The coordination involves hormonal signals like abscisic acid and cytokinin, which report local conditions back to the rest of the plant. This integration of spatial information is another facet of plant decision-making that resembles strategic planning. Even underground, plants engage in complex resource allocation calculus. Plants optimize leaf arrangement to maximize light capture. The arrangement of leaves on a stem, called phyllotaxis, is not random, but often follows precise mathematical patterns that minimize shadowing and maximize light capture. By positioning leaves at angles governed by the Fibonacci sequence, many plants ensure that upper leaves do not block light from those below. This pattern emerges from simple chemical feedback at the growing tip, where auxin gradients guide the placement of each new leaf primordium. The result is a remarkably efficient solar collection system, achieved without any conscious design. The geometry can be observed in rosettes, spirals, and other striking patterns found throughout the plant kingdom. This optimization is so effective that architects and engineers sometimes mimic it in solar panel arrays. plants have been solving light harvesting problems for hundreds of millions of years using elegant developmental rules. Fibonacci patterns in plants emerge from internal feedback systems. The beautiful spirals visible in sunflower heads, pine cones, and succulent rosettes are often arranged according to numbers in the Fibonacci sequence: 1, 1, 2, 3, 5, 8, and so on. These patterns emerge not from genetic blueprints, but from dynamic interactions at the shoot or flower meristem. Auxine accumulates in spots that become new primordia, and previously formed primordia inhibit auxine accumulation nearby, pushing the next primordium to form at a consistent angular distance. This self-organizing process naturally converges on the golden angle of around 137 degrees, which generates Fibonacci spirals. The mathematical elegance of these patterns arises from simple chemical rules, without any need for central planning. It is a striking example of how complex order can emerge from local feedback. Fibonacci phyllotaxis reveals that plants are masters of emergent geometry. Climbing tendrils test surfaces and adjust their grip accordingly. Tendrils of climbing plants like peas, grapes, and passionflower do not simply coil on contact with any object. They explore surfaces and evaluate their suitability for gripping. When a tendril touches a thin support of the right diameter, it coils tightly around it. If the object is too thick, smooth, or unstable, the tendril may release and continue searching. Coiling is driven by differential cell growth on opposite sides of the tendril, controlled by touch-sensitive signals. Once securely attached, tendrils often form spring-like helical structures that absorb shock and allow flexibility in the wind. This adaptive gripping behavior enhances the plant's ability to climb efficiently and safely toward light. The tendrils' testing and adjustment represent a tactile form of decision-making. It chooses its supports with surprising discrimination. Some climbing plants distinguish smooth surfaces from rough ones. Not all surfaces are equal in the eyes of a climbing plant, and experiments have shown that some species preferentially attach to supports with particular textures. Rough surfaces provide more grip for twining stems or adhesive roots, while smooth surfaces may be avoided or require different attachment strategies. Sensory cells in the tendril or stem detect the surface's microtopography and modulate growth accordingly. This discrimination helps plants avoid wasting resources on unsuitable supports that would not sustain their weight. The ability to distinguish textures again underscores the tactile intelligence of climbing plants. Their movements may be slow, but they are deliberate and informed by sensory feedback. Choosing the right support is a critical decision for any climber aiming for the canopy. Plants can alter stem thickness based on wind exposure. Plants that experience regular mechanical stress from wind often develop thicker, sturdier stems than those grown in sheltered conditions. This response, called thigmomorphogenesis, prepares the plant to withstand the forces of its environment. Mechanosensitive receptors detect bending and stretching in the stem, triggering hormonal changes that promote secondary growth and alter cell wall composition. The result is a more robust plant that is less likely to snap or lodge during storms. Trees grown in consistently windy locations tend to be shorter and thicker, while those in calm sites may grow taller and more slender. This plastic response distributes resources wisely, investing in strength only where it is needed. Thigmomorphogenesis is another way plants shape their bodies in response to experience. Seeds from stressed plants may inherit altered germination timing. When parent plants experience drought, heat, or herbivory, their seeds sometimes show altered germination behavior that reflects the mother's environment. This transgenerational plasticity can shift germination timing, dormancy depth, or sensitivity to environmental cues in the offspring. The effect is thought to be mediated by epigenetic modifications, changes in DNA methylation or histone marks that do not alter the genetic sequence itself. If the offspring's environment is similar to the mother's, inherited adjustments may improve survival by, for example, delaying germination until conditions improve. This inheritance of acquired information challenges the traditional view that Lamarckian inheritance does not occur. It also suggests a form of environmental memory spanning generations. Plants can thus pass on experiential lessons without words or conscious intent. This inherited timing can spread survival risk across generations. By adjusting the germination timing of their seeds in response to environmental stress, parent plants effectively bet hedge on behalf of their offspring. If all seeds germinated at the same time and conditions were poor, an entire generation could be lost. Staggered germination, whether across years or within a single season, spreads the risk of catastrophic failure. Stress-induced transgenerational effects may extend or increase variability in dormancy, ensuring that some seeds wait out unfavorable periods. This strategy is analogous to portfolio diversification in finance, reducing variance in long-term reproductive success. The information embedded in seed behavior reflects the experiences of the parent, creating a temporal inheritance of strategy. Through such mechanisms, plant lineages accumulate wisdom about their environments over many generations. Plant intelligence may operate on different timescales than animal minds. One reason plant intelligence is often overlooked is that it unfolds on timescales we find difficult to apprehend. Whereas animal decisions occur in fractions of a second, plant responses may take minutes, hours, or days. A root tip may take a week to decide which direction to grow, yet the decision is no less complex for its slowness. Time-lapse photography has revealed that plants are in constant, purposeful motion, simply too slow for us to perceive in real time. This temporal difference does not diminish the sophistication of plant behavior, it merely situates it in a different experiential frame. Understanding this timescale shift is essential for appreciating plant intelligence on its own terms. When we slow down our expectations, plant behavior reveals itself as intricate and deliberate. What appears slow to us may be deliberate in the plant world. From a human perspective, plants seem nearly motionless, but from the plant's own vantage, their movements may feel as purposeful and timely as our quick reflexes feel to us. A tendril searching for a support, a root tip navigating soil, or a flower tracking the sun all represent active engagement with the environment, just stretched across longer intervals. Evolutionary success does not require speed. It requires solving the problems of survival and reproduction effectively. Plants have been doing this for hundreds of millions of years, with remarkable success. Their pace may be an adaptation to their sessile lifestyle, where rapid movement is rarely advantageous. Judging intelligence by speed alone reflects animal-centric bias. Recognizing deliberation in slowness expands our conception of what intelligence can look like. Some researchers argue plants have a form of distributed cognition. Rather than localizing intelligence in a single organ, plants may distribute cognitive-like processes across their entire bodies. Every root tip, leaf, and meristem processes information and influences the behavior of the whole, communicating through hormonal, electrical, and chemical signals. This distribution resembles the architecture of neural networks or social insect colonies, where no single node controls the system. Proponents of this view describe plants as decentralized intelligences, achieving adaptive behavior through collective computation, rather than top-down command. Critics argue that calling this cognition stretches the term too far, but the debate itself has enriched our understanding of what minds are and where they might be found. Whether or not we call plants intelligent, their information processing is remarkably sophisticated. The distributed model challenges the primacy of the centralized brain. There is no central brain, but intelligence emerges from the whole plant. In animals, intelligence is typically associated with the brain, a centralized organ that integrates sensory input and directs behavior. Plants have no brain and no neurons, yet they exhibit adaptive behaviors that suggest intelligent coordination. The key insight is that intelligence can emerge from the interactions of many simpler parts, each contributing local decisions that aggregate into coherent whole-plant behavior. Hormones, electrical signals, and chemical gradients serve as the communication network that ties these parts together. The emergent character of plant intelligence means there is no single locus to point to, only a system whose capabilities exceed those of its components. This conception of intelligence aligns with ideas in complexity science and distributed computing. It invites us to see minds not as things but as processes arising from organization. This distributed model is inspiring new approaches to robotics. Roboticians have begun looking to plants for design inspiration, particularly for building machines that operate without a central controller. Traditional robots rely on powerful processors that gather sensor data and issue commands, but this architecture is vulnerable to single points of failure. Plant-inspired robots distribute sensing and actuation across their bodies, achieving robustness through redundancy and local processing. Such robots can grow, reconfigure, and explore environments in ways that rigid, centrally controlled machines cannot. The field of soft robotics, which builds machines from flexible materials, has found particularly rich inspiration in plant mechanics. By mimicking the decentralized control of plants, engineers hope to create robots that are adaptive, resilient, and suited for unpredictable environments. Nature's brainless organisms are proving to be powerful muses for cutting-edge technology. Engineers are building soft robots that grow rather than move. Inspired by the way plants extend their shoots and roots, engineers have developed robots that grow by extruding new material at their tips. Unlike wheeled or legged robots that traverse terrain, growing robots can navigate tight spaces, push through soil, or slip under rubble by adding length where needed. The growth is controlled by sensors at the tip that detect obstacles and adjust trajectory, mimicking the tropisms of plant roots. Some designs use pneumatic or fluidic systems to inflate and extend flexible tubes, achieving continuous forward progress without locomotion. These robots are well suited for applications like search and rescue, underground exploration, and medical procedures. Plant biology has provided a blueprint for a new paradigm in robotic design. What plants do naturally, engineers are now beginning to replicate in silicone and polymer. Plant-like algorithms help machines navigate without central control. The way plant roots forage for nutrients and water has inspired algorithms for distributed navigation in robotic swarms and network routing. Each agent or node makes local decisions based on environmental gradients, much like a root tip sensing moisture or minerals. When many agents follow such simple rules, the collective system achieves efficient coverage or routing without any central planner. These bio-inspired algorithms are robust, scalable, and tolerant of individual failures. They have found applications in searching disaster sites, monitoring environmental pollution, and optimizing data center traffic. The success of plant-inspired algorithms demonstrates that brainless navigation can be remarkably effective. Plants thus contribute not only to biology, but also to computer science and engineering. root growth strategies inspire underground exploratory robots. Exploring underground environments presents unique challenges: darkness, pressure, unpredictable obstacles, and the need to push through dense material. Plant roots have been evolving solutions to these challenges for hundreds of millions of years, and roboticists are now borrowing their strategies. Root-inspired robots use compliant, growing tips that sense resistance and change direction, avoiding obstacles rather than breaking through them. Some designs incorporate root-like branching to maximize coverage and sampling. The mechanical designs often employ soft materials that can deform and recover, reducing breakage risk. Applications include soil sampling, burrowing through rubble, and even extraterrestrial exploration on Mars or asteroids. Roots are the original underground navigators, and their strategies are now being translated into machines. Plants are being explored as living environmental sensors. Because plants are continuously sampling their surroundings through roots, leaves, and stomata, they are natural candidates for environmental monitoring. Researchers have proposed using plants as living sensors for pollution, pathogens, or toxins, with genetic modifications that cause the plant to produce visible signals, such as color changes, in the presence of target substances. This approach could provide low-cost, distributed sensing networks across farmlands, cities, or ecosystems. Plants already detect and respond to many chemicals we care about monitoring. The challenge is translating their internal signals into outputs we can read easily. Living sensors are self-powering, self-repairing, and biodegradable, advantages over electronic sensors. As biotechnology advances, plant-based sensing may become a routine tool for surveillance of environmental health. Living plant tissue can detect airborne toxins with high sensitivity. Plant cells possess molecular machinery that can detect extremely low concentrations of airborne chemicals, sometimes rivaling or exceeding the sensitivity of electronic noses. This sensitivity evolved because plants needed to monitor volatile cues from neighbors, attackers, and pollinators all at once. By engineering plants to couple detection to observable outputs, like fluorescence or pigment change, scientists can leverage this natural sensitivity for practical sensing. Demonstrations have shown plants that glow in the presence of explosives or change color when exposed to heavy metals. Such living sensors offer continuous, low-power monitoring without batteries or maintenance. The sensitivity and specificity of plant detection systems open exciting possibilities for homeland security, agriculture, and medicine. Plants may one day serve as sentinels, guarding our air quality. Some scientists have used plants as biological logic gates. In synthetic biology, researchers have engineered plants to perform simple logical operations by linking gene circuits that mimic electronic gates. An AND gate, for example, might require two chemical inputs to activate a fluorescent output, while an OR gate would respond to either input alone. More complex circuits can be assembled by chaining gates together, allowing plants to process combinations of environmental cues. These living logic gates are not as fast as silicon, but they offer remarkable advantages in self-replication and environmental interfacing. Potential applications include programmable crop responses, where plants only activate defenses or produce compounds when specific conditions are met. Living computation blur the boundary between biology and technology. Plants are becoming programmable platforms for bio-based information processing. Swarm robotics borrows principles from decentralized plant networks. Swarm robotics aims to coordinate many simple agents to achieve complex tasks without a central coordinator, much like ant colonies or bird flocks. Plants, through their root networks and mycorrhizal connections, offer another model of distributed cooperation. Swarm algorithms inspired by plant fungal nutrient sharing can optimize resource distribution among robots operating in resource-limited environments. The resilience of plant networks to local failures translates into robustness for robotic swarms facing damaged or lost units. By studying how plants allocate energy and information across distributed nodes, engineers develop swarms that self-organize efficiently. These bio-inspired systems are proving valuable for environmental monitoring, agriculture, and space exploration. The Wood Wide Web is becoming a design template for the next generation of collaborative machines. Collective plant behavior in forests resembles swarm intelligence. In dense forests, individual trees and the fungal networks connecting them can exhibit collective behaviors that benefit the community as a whole. Nutrient sharing, coordinated defense signaling, and synchronized masting all represent emergent properties of the forest as a system. No single tree orchestrates these outcomes. They arise from local interactions aggregating upward. This closely parallels swarm intelligence, where simple rules followed by many individuals produce sophisticated group behavior. The forest, in this view, is not a collection of isolated organisms, but a superorganism whose intelligence is distributed across countless participants. Such perspectives invite us to rethink the boundaries of individual and collective agency in nature. The forest thinks, in its own slow, distributed way. Desert plants coordinate flowering based on shared environmental cues. In harsh desert environments, many plant species synchronize their flowering in response to rare, favorable conditions like rainfall. This population-level coordination ensures that when flowers do appear, they bloom en masse, attracting pollinators that might otherwise be scarce. The cue is typically environmental, such as soil moisture or temperature thresholds, and is perceived independently by each plant. Yet the result is a collective burst of blooming that transforms barren landscapes into colorful spectacles. Such synchrony increases pollination success and seed set for everyone involved. It represents a form of distributed coordination without any leader or plan, a natural swarm of flowers. The desert's rare blooms are a testament to the power of shared environmental sensing. This population-level coordination ensures pollinator availability. Mass flowering events attract pollinators from across the landscape, providing a concentrated and reliable food source that isolated blooms could not offer. In return, the pollinators provide efficient pollen transfer among the synchronized flowers, boosting reproductive success for the entire population. This mutualistic coordination benefits both plants and pollinators, a win-win made possible by environmental synchrony. If flowers bloomed at random times, pollinators might miss many opportunities or be too dispersed to visit frequently. By aligning their reproductive efforts, plants effectively guarantee that pollinators will be present and attentive. This coordination is especially critical in unpredictable environments, where both plants and pollinators face resource scarcity. Population-level synchrony is a collective solution to the challenge of finding mates in a sparse world. Plants challenge our animal-centric definitions of intelligence. Most definitions of intelligence are built around characteristics found in animals: brains, neurons, rapid behavior, and conscious experience. Plants possess none of these, yet they sense their environments, integrate information, learn, remember, and respond in adaptive ways. If intelligence is the ability to solve problems and enhance survival, then plants qualify on every count. The Plant Challenge invites us to revise our definitions, rather than dismiss plant capabilities as mere automation. Expanding the concept of intelligence does not diminish what animals do. It enriches our understanding of the diversity of solutions life has evolved. Plants show that minds need not look or work like ours to be effective. Nature's experiments in problem solving are far broader than we once imagined. Intelligence may exist on a spectrum rather than as a binary property. The debate over plant intelligence highlights a broader question. Is intelligence a yes or no trait, or does it come in degrees? Viewing intelligence as a spectrum allows us to place different organisms along a continuum of cognitive complexity, from microbes to humans. Plants would fall somewhere between simple reactive systems and highly centralized animal brains, possessing forms of perception, learning, and adaptation that warrant the label intelligent to some degree. Such a perspective dissolves sharp dichotomies and invites appreciation of the many ways life has solved the problem of survival. It is not a matter of whether plants think like us, but of recognizing that they think in their own way. A spectral view of intelligence is both scientifically productive and philosophically humbling. Plants have thrived on Earth for over 400 million years. The earliest land plants appeared around 470 million years ago, and since then they have colonized virtually every terrestrial environment on the planet. Through mass extinctions, ice ages, asteroid impacts, and continental drift, plants have persisted and diversified into hundreds of thousands of species. Their success is a testament to their adaptive flexibility and robust survival strategies. Without plants, the atmosphere would lack sufficient oxygen, food chains would collapse, and complex animal life as we know it could not exist. Plants built the soils, shaped the climates, and provided the foundation for terrestrial ecosystems. Their longevity far exceeds that of any particular animal lineage, suggesting that their sessile, distributed mode of life is extraordinarily effective. Plants are among the most successful complex organisms Earth has ever produced. Their quiet success suggests hidden adaptive strategies. The fact that plants have outlasted so many animal lineages and survived multiple global catastrophes hints at deep adaptive advantages we are only beginning to appreciate. Their modular, redundant body plans mean that losing a branch or root does not doom the organism. Their decentralized information processing may be more robust than centralized nervous systems against sudden shocks. Their ability to enter dormancy and resume growth under favorable conditions allows survival through prolonged adversity. Chemical diversity provides layered defenses and adaptable responses to novel threats. Such strategies are not immediately obvious because plants do not move or vocalize, but they are powerful enablers of evolutionary success. The quiet persistence of plants is itself a form of wisdom. Studying plants may reshape our understanding of what minds can be. As researchers delve deeper into plant behavior, they are encountering phenomena that force a reconsideration of fundamental concepts like cognition, learning, and intelligence. The discovery of memory in plants, decision-making in roots, and communication through chemical signals challenges anthropocentric frameworks. A broader conception of mind emerging from studies of plants could transform philosophy, psychology, and neuroscience. It suggests that the boundaries we draw around consciousness and thought may be culturally contingent rather than natural. If plants possess even rudimentary forms of awareness, then minds may be far more widespread in nature than we assumed. This shift in perspective invites awe at the diversity of cognitive life on Earth. Plants may ultimately teach us as much about ourselves as about the botanical world. The question of plant consciousness remains philosophically open. While there is growing evidence that plants process information, learn and adapt, the question of whether they experience anything, whether there is something it is like to be a plant, remains unresolved. Consciousness is notoriously difficult to define, and even harder to measure, in organisms lacking brains. Some philosophers argue that consciousness requires certain neural architectures, while others propose that any sufficiently complex information processing might be accompanied by subjective experience. If plants do have some form of awareness, it would be utterly unlike our own, shaped by their stationary existence and chemical senses. This uncertainty is not a weakness of plant science, but a reflection of the deep mystery of consciousness itself. The plant question opens windows onto one of the hardest problems in all of philosophy. Plants may perceive the world in ways we cannot yet imagine. Given that plants sense light gradients, gravity, touch, humidity, chemicals, and perhaps electrical fields, their perceptual world is rich and multidimensional, though utterly alien to human experience. They may experience time differently, stretching over days what we perceive in seconds. They may feel the soil as intimately as we feel air on our skin. They may sense community and kinship through chemical whispers that have no analog in our sensory repertoire. Our current scientific tools capture only a fraction of what plants might perceive, and future discoveries may reveal dimensions of plant experience we cannot currently conceive. Imagining plant perception invites humility about the limits of human understanding. The green world remains partly mysterious, waiting for new questions and new methods. Every forest may be more interconnected than it appears. When we walk through a forest, we see individual trees standing separately, each a distinct organism. But beneath the surface, roots intertwine and fungal networks link tree to tree, passing water, nutrients, and chemical signals across the community. What looks like a collection of individuals is actually a superorganism, or at least a tightly coupled network, where the fate of one affects many. This interconnection has practical implications for forestry and conservation. Removing one tree may damage the network that sustains its neighbors. It also offers a metaphor for human society, reminding us that apparent separateness often masks deeper connections. Forests are not just scenery, they are living networks of mutual dependence. The quiet canopy conceals a humming web of life. Beneath your feet, a web of communication is quietly humming. Right now, wherever you stand on vegetated land, there is almost certainly a fungal network threading through the soil beneath you. This mycelium is exchanging signals with plant roots, redistributing nutrients, and responding to changes in the environment. The network may connect trees separated by meters or even tens of meters, forming a living internet of the forest floor. Most of this activity is invisible and silent to human senses, yet it is continuous and essential for ecosystem function. The hum of this underground web is a metaphor for the hidden complexity underlying familiar landscapes. We walk on a living membrane of interconnected organisms, oblivious to the conversations happening below. Awareness of this hidden world deepens appreciation for the ground beneath our feet. The green world invites us to see intelligence in a new light. Plants have been Earth's quiet geniuses for hundreds of millions of years, solving problems of survival without brains, muscles, or mobility. Their success forces us to reconsider what intelligence really means and where it can be found. If intelligence is the capacity to sense, integrate information, and respond adaptively, then plants are intelligent by any reasonable measure. Recognizing this broadens our appreciation of life's diversity and deepens our humility about human uniqueness. The green world invites not just wonder, but a shift in worldview. seeing minds not as rare accidents, but as emergent properties of living systems everywhere. Perhaps intelligence is less about having a brain, and more about being embedded in the world, connected, responsive, and endlessly adaptive. Plants embody this quiet wisdom, and they invite us to learn from their example. And now the quiet wonders of plant intelligence begin to gently fade into the stillness of the night. You have wandered through over 100 peaceful truths about the hidden awareness of the botanical world. From leaves that fold at the slightest touch to roots that navigate the darkness with purpose. From chemical warnings drifting through the forest air to fungal networks connecting entire communities of trees. You have explored how plants sense their surroundings, remember past experiences, and make decisions shaped by context and history. You have learned that beneath the stillness of every garden and forest, there is constant communication, adaptation, and a form of intelligence we are only beginning to understand. You are connected to this story more deeply than you may realize. Right now, as you listen, plants all around the world are quietly sensing, responding, and adapting. They are providing the oxygen in your lungs and the food on your table. They have been doing this work since long before humans walked the earth, and they will continue long after tonight. If your eyes feel heavy now, let them close. Let the rhythm of your breath become steady, like the gentle pulse of sap rising through a stem, like the quiet patience of a seed waiting for spring. Tonight, as you rest, the plant world continues its timeless work. Photosynthesis will pause with the darkness, but roots will keep exploring. Chemical signals will still flow through stems and leaves. Fungal networks will carry messages between trees standing silent under the stars. You are a small, warm presence within this vast green world, peacefully resting among the wonders. Sleep well, sweet dreams. And may you carry with you the quiet knowledge that intelligence takes many forms, and some of the wisest beings on earth are rooted gently in the soil. Thank you for joining me on this journey through the mysteries of plant intelligence. If you enjoyed this video, please subscribe to Drowsy Discovery for more soothing explorations of how the natural world quietly thrives around us. There are more wonders waiting, more secrets hidden in leaves and roots, more gentle truths to drift through on quiet evenings. Rest well, and may the patient wisdom of the plant world keep you company through the night. This has been Drowsy Discovery. Good night.